So let's check the antigenicity of the protein. Now, what is antigenicity? Antigenicity or antigenic reactivity refers to the capacity of viruses to bind to specific antibody molecules. Okay, its capacity to bind. Now, antigenicity is the ability to be specifically recognized by the antibodies generated as a result of immune response to the given substance, right? So here, let me just show you two softwares to check antigenicity. Okay, there are different softwares, just that you need to go with the protocol. That's about it. In every software, there'll be something called as a help button, where actually they'll be completely referring all the steps that you need to follow, why you are doing that process and stuff. Okay, but I'm just giving you a practical exercise so that you can use these two softwares as well. Okay, so let's go to the first software, that is Vaxigen 2.0. Okay, so go to this site. Okay, sorry, go to this link. Okay, just copy this link and then paste it in your Google, then you will be redirected to the software. It's called as Vaxigen 2.0. So now what I did was I searched for Vaxigen 2.0 and the first result that you see. So this is what you need to take. Okay, so let's take it. Okay, so this was the link I was mentioning you about. Okay, so here is your home page of Vaxigen. Okay, now see the boxes. Enter a protein sequence here, plain format only. So here what the first step that you need to do is paste the copied protein sequence, which we already have. So it's saved as protein 1 spike glycoprotein. So let's copy it, Control C, and paste it here, Control V. See, this is what I've mentioned. If you click the help button, you will see what Vaxigen does. It's just an overview of it. Okay, yeah. So either you can paste your sequence, or you can choose file and then upload your notepad file or whatever, in which format that you have uploaded or you have saved it. Okay, so now we have pasted the copied protein sequence. Now our target organism, which we already know, it's virus, right? Target organism. So it's virus. Now if you want to give a threshold, up to you, you can give, but I'm making it as default. I'm not giving any threshold. Let the software decide. Okay, so I'm just selecting a target organism and clicking the submit button. So here you have multiple options like ACC output. It's the different output mode. Okay, summary mode. You can try all these things. I am making it as a default sequence output, because what I want is the output of this sequence. Okay, when you select this, again all the protein sequences will go off. So do this again if this issue appears. Okay, so I've copied the sequence, selected the target organism, and I am gonna click the submit button. Okay, before that, let's just look at Vaxigen help page. Okay, it's the same thing. See, when you scroll down, there are other prediction servers. When you click there, okay, you can't see anything. Anyway, okay, let's continue with our step. Virus and then submit. Then you'll get your result. Here in the results page, you know that the model selected is virus, and the threshold for this model that they have set was 0.4. Okay, this was our sequence, right? Next is what is needed. Overall prediction for the protective antigen is 0.5647. which means it's above its threshold, right? So which means what it's considered as an antigenic protein. So that's why they have mentioned probable antigen. So if it's an antigenic protein, then only we can design a vaccine for it, right? Because it has to have an ability to bind to a specific antibody molecule. That's what our aim is. Okay, so now we can be happy by saying that this protein sequence can be moved forward with the next step. Okay, but let's do this with another software also, okay, and see if it's a probable antigen in that software also. It's always good to check for multiple softwares and then see our result is replicable. So let's open another software of immunomedicine group, okay, to check the antigenicity. Let me show you the slide of it, okay. So this was what we found, right? So it was probable antigen in the result, okay. So if it's above the threshold, if it's an antigen, then it is considered as an antigenic protein. Next, another software of immunomedicine group. So you just have to go to this particular link, okay. So I have searched it as immunomedicine group antigenic tool, and then let's go to this result, okay. Tools predicted antigenic peptides, or just copy and paste the link, okay. So this is your site. If you want any directions, you can go and see this page where they'll be mentioning about it, about the introduction of it, about the antibodies and the softwares, okay. But let's go with them. So if you want to enter a short name for your sequence, yeah, let me just give SG, okay. It's a spike glycoprotein. I'm just giving a short name. That's just optional. So yeah, replace the example sequence with your protein sequence. So just delete it, and then you just have to paste your sequence right here, okay. So you don't have to do or paste the FASTA header, okay. So delete it and paste just your plain format sequence, and then click submit, okay. So the result is loading. Okay, so now you can see the result page, okay. So here you'll actually get the length of your sequence, okay. Let's just wait for the results to be loaded. Okay, so as you can see, the length, right, the length of the sequence is mentioned. It's 208 residues long. Then you have the average antigenic propensity, okay, which is 1.0543. That's the antigenic propensity. Next, you have the antigenic plot for the sequence SG. This was the short name which I've given, right? So let's look at the graph. You can see what this graph means. Okay, let's just take the 150th position. As you can see, this is the sequence number. So in 150th sequence number, or in 150th position of the amino acid sequence, which is 208 residues long, which has 208 residues, because the red line, it shows still 208, right? So in 150th position of the protein, there is increased antigenicity, which means the y-axis represents the average antigenic propensity, okay. That is about 1.2, okay. That's the maximum, 1.2. And then in 100th position, it's between 1.15 and 1.2. So here what they have taken is, they have taken the average of it, and then the average antigenic propensity is 1.0543, okay. So in this software, the average antigenic propensity is this, okay. But here we don't have the threshold so that we can compare it, okay. I would say the first software is better than the second one, but here we can always see the graph. But by these methods, efficient vaccines can be developed, okay. So now, as you can see, the results, it was 1.0543, right? Even though the threshold is not mentioned. In the first software that we have checked, it was 0.5647. So you can see that the results are different, right? The results are different in both servers, probably because of the difference in threshold value. Here they have mentioned that they have made the threshold as 0.4. Here they haven't mentioned the threshold. Okay, but yes, now similarly your task is to note about all the 19 protein sequences that you have saved in different notepad files. Just do all the 19 proteins antigenicity, and then in an Excel sheet, okay, just go to an Excel sheet, and then in the first column write the protein number, since you have already mentioned or saved in the notepad as protein 1 to 19, and this one would be your first one, the other 19, so you will be having a total of 20 proteins, right? So do one thing, you make an Excel sheet, and then in the first column write protein number, and then 1 2 3 followed by it, and in the second column write the Vaxogen antigenic score. So here it was let me just take the Vaxogen side, it was 0.5647, right? This was, so write it for the protein 1, write it as 0.5647, and in the third column write the other the Vaxogen antigenic nature, which is probable antigen. That's what it is mentioned, right? So write it as probable antigen. Then in the fourth column write the immunomedicine antigenic score, which was 1.0543. So you'll be having 4 columns. Okay, now I missed to mention one more thing which is there in immunomedicine group. It says that there are 10 antigenic determinants in your sequence, which means it's a probable antigen. It's an antigen. You don't have to worry about it. See here, they have mentioned the fourth position, 27. Then in 74 you have, so 74 may be in this position. Okay, then you have 94th in 94th, then 110th position, 133 sequence number, 142. Okay, this is your start position, and this is your end position from 191 to 197. Okay, that would be probably here 191 to 197, which means this is one antigenic determinant, and even the sequence is mentioned, FGITVQY. Okay, so even that is mentioned. So you can see that, or you can say that in your antigenic protein sequence you have 10 antigenic determinants. Even though threshold is not mentioned, the antigenic determinants and the graph is beautifully plotted. So this is how you need to compare the softwares also. For the threshold, you can always say this is the threshold, and so here in this result you can say that this is the threshold and it's a probable antigen. You can also add your information, as in my sequence I have 10 antigenic determinants in all these positions. Okay, that's why I've mentioned to try out more softwares. Okay, so that you can see you can check whether if you can reproduce your results. At the end of the day, it says it is antigenic. That's what we need. Let's say this sequence says that it is not antigenic. If it's non-antigenic, then you may have to try, you have to try another protein sequence. Okay, so as I've mentioned, is you need to make an Excel sheet. Just name the Excel sheet as you wish. Open the Excel sheet. Okay, and then write as I've mentioned. Here what I have made is, I have written it as protein number, then I've made a heading as antigenic prediction, where since we have used two softwares under that, Vaxogen and immunomedicine. So under the Vaxogen you have antigenic score and antigenic nature, right? In antigenic nature it was mentioned it's a probable antigen. So I'm gonna start with the protein number. So it was the number 1, which was spike glycoprotein, right? I'm just writing it as SG. So similarly you can also write your protein numbers. Okay, if you have just written protein number as 1, yeah, you can very well do that. Now antigenic score, it was 0.5647. Okay, so copy this and paste it here. In the immunomedicine, it was 1.0543. Now paste it here. Okay, so these were the things that we did. Okay, so as of now, we have retrieved the protein sequence and determined its antigenic propensity or antigenic nature of it. Now the third step that we are gonna do is to predict the allergic nature of the antigenic protein. That's what our third step. It again comes under the prediction of functional properties. The first property was antigenicity. The next is allergenicity.